One of the more uncomfortable questions in the NMN supplement space, uncomfortable primarily for the companies selling premium NMN products, concerns what actually happens to an NMN molecule between the moment you swallow a capsule and the moment NAD+ appears in your cells. The answer, based on current biochemical understanding, involves a metabolic journey with some significant stops along the way. And at least one of those stops raises the possibility that you're paying a substantial premium for a roundabout route to the same destination that a cheaper molecule reaches more directly. NMN is a relatively large molecule that cannot simply diffuse across intestinal cell membranes. Research has shown that a substantial portion of orally ingested NMN is converted to nicotinamide riboside (NR) by an enzyme called CD73 in the gut lumen before it enters intestinal cells. From there, NR undergoes further conversion, eventually producing nicotinamide (NM) as an intermediate. NAM then feeds into NAD+ synthesis pathways that are essentially identical to the routes niacin uses after its own enzymatic processing. The molecular paths converge at the point of actual NAD+ production. What this means in practical terms is that the more direct pathway that NMN marketing describes may not be as direct in reality as it is in diagrams. The claim that NMN bypasses degradative steps that other precursors can't avoid is, at best, an oversimplification of a complex and incompletely understood set of intestinal and hepatic metabolic processes. Some tissues may handle NMN differently than the gut does. There is evidence that some cell types can absorb NMN directly via specific transporters. But the idea that swallowing NMN guarantees a more efficient or more complete NAD+ elevation in all tissues compared to niacin is not established by current human evidence. There are legitimate tissue-specific differences worth acknowledging. The liver processes NAD+ precursors differently than muscle does, and muscle metabolizes them differently than brain. Whether these tissue-specific differences translate into clinically meaningful advantages for NMN over niacin in any particular organ system is an open research question. The honest answer is that we don't know with enough precision yet to justify the cost differential on mechanistic grounds alone. What would be most useful for consumers navigating this space is a personal biomarker approach. Test your NAD+ levels on niacin alone, compare to NMN alone after a washout period, and evaluate your individual response. This kind of structured self-experimentation, imperfect as it is, provides more actionable personal data than any general pathway diagram. The principle that more complex and expensive doesn't automatically mean more effective applies here with particular force. If the metabolic bus, niacin, gets you to the same destination as the molecular Uber, NMN, the relevant question is whether the price of the Uber is justified by any measurable difference in arrival quality.